The role of iron in cellular function, a comprehensive analysis. Introduction. Iron is an essential micronutrient for the proper functioning of numerous biological processes in the human body. From oxygen transport to energy production, iron plays a critical role in various physiological functions. In order to maintain iron homeostasis, several intricate molecular mechanisms have evolved to regulate iron uptake, storage, utilization, and export. In this comprehensive analysis, we will delve into 12 key aspects of iron metabolism, ranging from the molecular basis of iron overload disorders to the impact of iron on immune function. By exploring these topics, we aim to gain a deeper understanding of the intricate network that governs iron metabolism and its implications in health and disease. The intricate regulation of intracellular iron levels primarily relies on the activity of iron regulatory proteins, IRPs. These RNA-binding proteins exert control by binding to specific sequences known as iron response elements, IRES, located in the mRNA of several genes involved in iron homeostasis. The binding of IRPs to IRES can impact gene expression at the post-transcriptional level, ultimately influencing iron uptake, storage, and utilization. This topic explores the structure and function of IRPs, their interactions with IRES, and their role in maintaining the delicate balance of iron within cells. One of the essential players in iron uptake and transport is the transferrin receptor, TFR. This membrane-bound receptor binds to transferrin, the major plasma protein responsible for the transport of iron. Through receptor-mediated endocytosis, TFR internalizes iron-loaded transferrin complexes into cells. The subsequent release of iron from transferrin enables its utilization in numerous vital processes. This topic delves into the structure, regulation, and physiological significance of the transferrin receptor in maintaining iron homeostasis. Central to the regulation of iron metabolism is the hormone hepcidin. Produced predominantly by the liver, hepcidin plays a crucial role in modulating iron levels through its interaction with the iron exporter protein, ferroportin. Upon binding to ferroportin, hepcidin promotes its internalization and degradation, thus reducing iron export from cells. By exploring the intricate regulatory pathways involving hepcidin, this topic sheds light on the key mechanisms governing systemic iron balance. Iron-sulfur, Fe-S, clusters are versatile cofactors critical for the function of numerous enzymes and proteins involved in essential cellular processes. From electron transfer in respiratory chains to DNA repair and replication, Fe-S clusters contribute to various metabolic pathways. This topic explores the biogenesis and intricate assembly processes underlying the generation of functional Fe-S clusters, elucidating their significance in cellular function. The process of iron uptake from the extracellular environment varies among different cell types and tissues. This topic focuses on the intricate regulatory mechanisms that govern iron uptake into cells. It explores the involvement of various membrane transporters, such as DMT1 and ZIP8, and their regulation by iron-responsive elements and transcription factors. The topic also examines the impact of heme and non-heme iron sources on intracellular iron levels. Efficient iron export from cells is crucial for maintaining systemic iron balance. This topic explores the key molecular players involved in iron export, with a primary focus on the iron exporter protein, ferroportin. It delves into the mechanisms regulating ferroportin expression, post-translational modifications, and interaction with hepcidin. Additionally, this topic examines the role of other proteins, such as ceruloplasmin, in facilitating iron export. Mitochondria, the powerhouses of the cell, harbor numerous iron-dependent processes essential for cellular energy production. This topic provides an in-depth analysis of the intricate relationship between iron and mitochondrial function. It explores the role of iron in respiratory chain complexes, iron-sulfur cluster biogenesis, and the influence of impaired iron metabolism on mitochondrial diseases. Transcriptional control represents a significant mechanism by which cells regulate iron metabolism. This topic investigates the role of iron-regulated transcription factors, such as hypoxia-inducible factors, HIFs, and iron regulatory proteins, IRPs, in modulating the expression of genes involved in iron homeostasis. The intricate crosstalk between regulatory elements, iron-responsive elements, and specific transcription factors is explored, shedding light on the complex network that governs iron-dependent gene expression. In order to ensure efficient and targeted delivery of iron to its various cellular destinations, specialized proteins known as iron chaperones and trafficking molecules participate in the regulation of iron transport and distribution. This topic unravels the roles of key chaperones, such as poly-RC binding protein 1, PCBP1, in coordinating iron trafficking and its delivery to specific cellular compartments, including the nucleus and mitochondria. Iron overload disorders, such as hereditary hemochromatosis, encompass a group of genetic disorders characterized by excessive iron accumulation in tissues. This topic delves into the underlying molecular mechanisms leading to these disorders. It explores genetic mutations affecting iron homeostasis, such as the HFE gene mutation, and the impact on iron absorption, storage, and regulation. Additionally, the topic discusses diagnostic approaches and treatment strategies for iron overload disorders. Iron plays a pivotal role in maintaining proper brain function, with disruptions in iron homeostasis linked to various neurological disorders. This topic examines the intricate mechanisms of iron regulation specific to the brain. It explores the blood-brain barrier, iron transport across brain cells, and the involvement of brain-specific iron regulatory proteins in neuronal health and disease. Beyond its vital role in metabolism, iron influences immune function and plays a crucial role in host defense against pathogens. This topic unravels the intricate interactions between iron and the immune system. It explores the influence of iron availability on immune cell differentiation, function, and regulation. The effects of iron dysregulation on inflammatory diseases, infection susceptibility, and the potential therapeutic implications are also discussed. In conclusion, the comprehensive analysis presented here sheds light on the intricate web of molecular mechanisms governing iron metabolism. From the regulatory functions of IRPs and TFR to the role of hepcidin in maintaining systemic iron balance, each topic explored provides a deeper understanding of the interplay between iron and cellular function. The molecular basis of iron overload disorders, the impact of iron on brain and immune function, and the complexities of iron-sulfur cluster biogenesis further emphasize the profound importance of iron in human physiology. By unraveling these intricacies, we pave the way for future research, novel therapeutic interventions, and a deeper appreciation of the fundamental role of iron in health and disease.